Hello everyone, my name is Evanthia Koukouli and I am a PhD student at Lancaster University. Today I'm presenting a joint work with Juyun Park, Frank Dondellinger and Dennis Wang with the title A Regularized Functional Regression Model Enabling Transcriptome-wide Dosage-Dependent Association Study of Cancer Drug Response. Cancer is a heterogeneous disease with individual tumors showing sometimes very different mutational and molecular profiles. As a result, anti-cancer treatment failure rates are often high, motivating the need for more personalized treatment strategies. At the same time, we know that it's not only the drug selection that plays a major role on patient remission, but also the drug dosage. Genetic factors can be used as predictive markers of drug response and can help in fine-tune the dosage for individual patients. In this work, we used cancer cell line screen data from the Genomics of Drug Sensitivity in Cancer project to build a dose-varying regression model which allows to estimate dose-dependent associations between anti-cancer drug response and gene expression at baseline. Our application focuses on 5 BRAF-targeted drugs used for the screening of more than 900 cancer cell lines. Microarray gene expression data, including more than 70,000 genes, was also used to incorporate the genetic information into the model. To estimate dose-dependent associations, we built a dose-varying coefficient model, and to identify the genetic markers that are associated to drug response, we applied a two-stage algorithm. This algorithm combines two steps. First, a variable screening step, where we rank the genes according to their utility, which is computed after marginally fitting the model for each gene. And the second step, a penalized regression step, where we further reduce the number of predictors by applying a group cut penalty. In our application, we detected 230 genes associated with a drug response. Most of the examined drugs had similar or worse behavior at all dosages to the dabrafenib drug, except from the AG664-1 drug, which showed better efficacy at higher dosages. We also observed a greater dabrafenib efficacy for cell lines originated from the endocrine system, autonomic ganglia, and hematopoietic and lymphoid tissues at lower dosages. We were also able to identify and to estimate the expected survival rates of the cancer cell lines for each drug examined, given their gene expression information at baseline, and we were also able to predict drug efficacy at untested drug dosages. We also found that identified genes were grouped into known cancer pathways, including MAPK signaling. We interestingly also found genes significantly overlapping with gene sets overexpressing an oncogenic form of KRAS gene. Predictive performance evaluation showed also strong and robust performance when it comes to predicting sensitivity to the administered drugs at specific dose intervals. We finally implemented the algorithm to cell lines showing some mutation in resistant mechanisms of BRAF. From this analysis, we found 65 genes, from which 42 were linked to either the BRAF gene, tumor genesis, or cancer treatment. In figure 4 of the poster, you can see the protein-protein interaction network between the selected gene set and the BRAF gene. To conclude, our approach can quantify those varying drug, gene, and cancer type effects on the cell line response. It can also reveal a number of unknown potential relationships between genetic characteristics and drug efficacy, and it is flexible enough to allow each drug to be examined separately or to allow for enrichment with additional covariates, for example, drug chemical information. You can find our full manuscript as a preprint online at the bioRxiv by scanning the QR code in the poster. Thank you very much for listening, and I hope you found my talk interesting.